Okay, so for those of you who were not in the lab today, for lab 9, let me just go through a couple things so you kind of are clear on what you'll need to do for today. So the first section here is a couple of microscope slides, and I have pictures of these on Canvas, and I will take a second at the end of this video to maybe go over a couple of details on what you should be drawing for that. The first one is just going to be a cross section. So if you see long strings of muscles, you're looking at the longitudinal section down the long way of the muscle cells. So you want to be looking at pouches or pockets. So you want to be seeing those fascicles and then the cells inside the fascicles for that cross section. And don't forget to label those two things on there. The second one will be motor neuron endings. So it's really cool that you can actually see the axons coming in. So the nerve coming into the muscle and how it integrates and creates these motor units at each of the cells. So the next part is going to be this just YouTube video, which I have posted on Canvas as well on that same page. So you should be able to pretty quickly go through that and answer these questions. But then the activity that you won't be able to participate at all in the actual doing of this activity is this electromyography using the BIOPAC equipment. However, I will need you to download the BIOPAC software to do the really quick analysis portion of this, which will be pretty simple and straightforward. So I am going to show you now a video that Dr. Maroff and I performed in the lab with the BIOPAC software to show you kind of how we actually went through to do that. And then I'll go through a little bit of the analysis and how you can fill out this table on your own. And then at the very end, I'll go through just a few of these questions. So I'm going to try to keep this as short as possible so that you guys can get through this fairly quickly. But I'll go through a few of these more difficult questions, just so that for those of you who weren't in class, you get the same experience as everybody who was there. I went through numbers 1 through 4 for the people who were in class, because those are kind of the more difficult questions in this lab. So let's take a look at the BIOPAC exercise and how that works. Okay, so we are going to be doing a BIOPAC exercise this week for part of the lab. So for today, we're doing something with grip. And so we have some equipment in the lab that looks like just what you see in these pictures right here. So we have a hand grip that the computer is going to use, is going to be recording our strength of our grip with some electrodes, which we have on the screen here. And so we have our subject set up with 3 electrodes on their arm, and that is going to measure how strong their grip is as we go through this. And it's going to be measuring the grip strength by measuring the electrical activity. So remember that muscles have to have nerves firing. So we have these motor nerve endings, which you'll see in the lab today as well. And so by firing messages through those nerve endings, we're going to be able to measure that electrical activity in the actual muscles themselves. So if you have more muscle activity happening, that probably means that you have more muscles being activated, more muscle fibers being activated. So we'll see how that looks. And so we're going to use something called a hand dynamometer today, and that's what this black thing is. All right, so we have our subject already in a seated position, and we have gel on their arm with the electrodes, so we can actually get a good electrical signal coming through there. And so we are going to have them with the dynamometer clench with their dominant hand for as hard as possible with for 2 seconds, just to calibrate the system. You want to pick it up so we can calibrate. Clench for 2 seconds and then relax. Okay, so that was just to calibrate the system. Once that's done, then we're going to just simply go through the exercise as it shows on the screen here. Okay, so what we're going to have our subject do is do a series of increasing strength in their grip series. So we have them holding the dynamometer in their dominant hand right now. And so they're going to try to start around 5 kilograms of force. Is it kilograms? Kilograms of force. And they're going to try to increase by 5 increments of kilograms in each subsequent clench that they do. So we're going to hit record and do their first one. So they're going to try to hold it steady at 5 kilograms per force. Then they're going to try to go up to 10 kilograms per force. And they're always going to be trying to clench for 2 seconds and then release for 2 seconds. And the screen follows you as you go throughout the exercise, so you don't lose any of your data. It just continues to record, and they go up as much as they can.
That's my max.
And so whenever they hit their maximum, then you can hit suspend. Okay, so once we're done with collecting our data, we go back through, scroll through, and just make sure that our data looks like it should. So this is some example data down here, and our subject's data does look like it needs to, so we can go ahead and continue through the module. Okay, so this next series is going to be still with the dominant hand, and our subject's maximum was not quite 25, around 22 or 23. So if we go over to tasks, we can see what they need to do for this exercise. So they're going to clench their hand dynamometer as hard as possible, maintain that force, and they're going to continue to hold that force until it has decreased by about 50%. So if their max was around 22, they would have to hold it until they get down to about 11, and then they can go ahead and release. So we're going to have them do that now. So you can see that their force started right around 23 or so. So they're going to try to hold it until it gets down to 11 or 12. So this is a fatigue exercise. We want to see how long they can hold and sustain this movement. So you have muscles firing in your forearm as this happens, but of course, as that is occurring, your muscles are using ATP, they're using energy that they need, and it can't be replenished constantly as these muscles are firing. Eventually, you're going to get to a point where you can't hold that grip strength any longer. They're grimacing, the subject is tired. About down. Okay, so once they've reached 50%, you can hit suspend, and you should see a gradual decrease, which we do see throughout the entire series. Okay, so as long as your data matches what the example data looks like, which it all should for the most part for this hand grip exercise, you can continue to move on throughout. It's always going to save your data for you, as long as you're just using the record and suspend buttons. As long as you're not exiting the entire exercise, it'll save your data for you. Okay, so we now have electrodes attached to the non-dominant arm, which is the subject's left arm, and we're going to go through the same exact steps again. You can always go and click on tasks to see what you need to be doing. So again, this is those 2-second cycles of clench and release, trying to increase the kilograms by about 5 every single time. And you might anticipate that for your non-dominant hand, we probably won't have as much grip strength because you just don't use that hand as often. And so you probably don't have as much muscle in your non-dominant hand, but we'll see. Okay, so that was their maximum. Again, we see the stair-step pattern with their data, so we can go ahead and click continue. And again, we're going to do the fatigue exercise on the non-dominant arm. Should have given me more of a break. We just jumped. We kind of jumped the gun there.
Notice how much faster my non-dominant is going down here. I don't have as much stamina.
We'll be able to compare this data afterwards, but you can clearly see decreasing quite a bit faster. And they started just over 20, so we'll wait until they're down around 10. And Dr. Maroff is glad that she's not being recorded right now for this, her facial expressions. All right, we'll click suspend. Again, decreases gradually throughout because this is just a fatigue exercise. So we'll go ahead and, so at this point, we've completed all that we need to for today. We did dominant and non-dominant arms. We did the increasing stair steps and then the fatigue exercises. So since we did everything and everything looks good, we can go ahead and click stop. And we're going to save that file and then we'll be able to analyze it now. Okay, so once you're all done, you will be able to hit done for the lesson because we're not doing the sound part. We can't use the headphones. And then we can go and analyze current data file, or you can save it to the computer to analyze it later. But if you're in the lab, you'll be doing this. At home, I'm going to give you some example data, and you'll be able to actually do this next part on your own. You can always drag down on the journal so you can see more of your data up here. Okay, so just at a first glance, if we look at this data, we have two sections here, one in this blue line and then one in the green. The green is the electrical force, so millivolts per second, not the electrical force, the electrical activity in the arm measured through the electrodes. And this is the force in kilograms measured through the hand dynamometer. But these should really reflect each other because the amount of electrical activity is going to be a reflection of how many muscle fibers you have active at that time. There are a few tools that you can use to analyze your data because you'll want to analyze each individual section at a time. So if you use this magnifying glass over here, you can zoom in on a particular section. And then if you, let's see, display and auto scale waveforms, that will make sure that everything is fitting within the graph so you can see all of your peaks as you need to be able to do that. You can also just hit Command Y to do that. So auto scale waveforms, and you should be able to then highlight individual waves in your data to be able to analyze each one of those. Okay, so there are a few tools that you'll need. This analysis is going to be pretty simple, but you're going to want to, for each of those stair steps of the increasing values, you are going to want to just highlight a section where it's pretty flat line at the top of that peak. You don't want to highlight the entire thing because then it's just going to be taking an average of all of this lower activity here. So we just want to know at each of these increments how much force was happening and how much electrical activity was occurring there. So if you highlight a little portion where it's flat, you can go up here to the top, and the blue line will correspond with the blue box, the green line with the green box, and you can just write down your mean kilograms of force right there. So this would be 14.7. And then the amount of electrical activity, again, just take the mean in millivolts per second. So that's going to be 0.129. So you can do that for each one of those. And then, okay, and so then for the fatigue exercise, so we want to see how long, what is it, the average force of the...
Yeah, how long it takes to get to fatigue, or 50% of our force.
The amount of time.
Yeah.
So if we want to know the amount of time that it takes to get fatigue, we want to be able to highlight the entire thing. So you can zoom back a little bit. If you go to display, zoom back. And we'll zoom out a little bit more. And again, you can just do auto scale waveforms to be able to view that. And I'm just going to highlight this entire section. Okay, so once you have the entire thing highlighted, you can get the total time that it took them to fatigue. So that is going to be the delta T value right here. So that's the total time for the portion that you have highlighted. You'll also want to get the peak, the maximum strength of that. So the maximum clench force you can just get by highlighting the first part of that grip strength exercise. Okay, so let me show you a couple of things now on my computer just so you can see a little bit better how to use the BIOPAC software. So you'll download this from the internet, and again, you just have to make an account, but you don't need to pay for anything. It's free for students to use at home on your computer if you use the link that I sent you. They won't send you like spam emails or anything. The app looks like this once you have it downloaded, so go ahead and open up the BIOPAC software, and it automatically will have the correct thing selected. So you'll just do analyze your own data, hit OK, and you just need to find a file on your computer. So if you have downloaded these already from Canvas, the male data and the female data, find wherever you have saved them. So if I open up the male data, you should see all of the stuff that we went through in lab, and you'll be able to copy and paste these answers into your Word document into that portion. The only tools that you need, again, I showed you this in the last portion, but I know it was hard to see on that computer. So just the magnifying glass to zoom in on your area, hit Command Y to auto scale waveforms or go up to display auto scale waveforms. And then you can highlight with the I-beam cursor each segment of your graph that you want to analyze. So if it's kind of messy, so we have a, it's kind of hard to maintain that grip at the certain level. So just choose a flat part of the graph, and from that, it'll highlight your force in kilograms as well as your electrical activity. And then you'll just write down the mean for your force in kilograms, the mean of your electrical activity in millivolts per second. And then when you get to the portion where you're going to analyze the fatigue and how long it took them to fatigue, you're going to highlight the entire thing until they obviously started to release their grip. And then you are going to use this delta T value. So it'll be in seconds alone. So that's just going to be how many total seconds were they able to hang on to that, the hand grip until they hit that 50% fatigue level. Okay, so once you do that, you'll just copy and paste those values into this table. So, for the male one, if we highlighted this portion, it should be about 8.95 and 0.117. So I just copied those into this Word document right here, and you'll do the exact same thing for the female data. So you'll get male and female, and then you'll be able to kind of compare the data, see how it changes between genders, and you can then answer some of these questions below here. Let me go through a couple of these. So when you hold an object, does the number of motor units remain the same, and are the same motor units used for the duration of holding an object? Well, if I hold up this coffee cup and I'm holding it completely stable out in front of me, is the number of motor units going to remain the same, go up, or go down for the duration of me holding it here? It is going to stay the same because the force that is needed for me to hold this mug is always going to stay the same, as long as I'm not trying to lift it up or let it fall to the ground. The number of motor units remains the same because the number of motor units is a reflection of the force. So the more motor units you recruit, the more force that you have. However, even though this object's not that heavy, eventually the muscles that I'm using to hold it will start to fatigue. But I'm not requiring the entire muscle in my bicep and forearm to be working because I'm not using my maximum amount of force to hold this object because it's not that heavy. So when certain muscle fibers or muscle cells start to fatigue, they can just kind of pass on to another cell and say, hey, you take over for a while. So you have this trade-off happening. So when certain cells fatigue, they just allow other cells around them to start to do the work for them. Okay, the relationship between muscle tension and load. Well, me holding this object is creating a force, and we call that force load. My arm is creating a force to hold it in place, and we call that force tension. So load, tension. And so if I'm holding an object still, those two forces are going to be equal, right? Because that is what is allowing me to hold this object in place. Now, if you are going to push a door open, pull something, pull the trash out of your trash can, or lift your body up in the morning when you go to stand up, you need just enough tension in your muscles to overcome the load. So think about a stable object, or if you are trying to work against like pushing a door open, because it will be a little bit different. It's either going to be equal or it's going to be just enough tension in your muscle to overcome the load. Okay, differences in males and females. Yes, we see differences, and that's simply because males are going to have more muscle. So their muscles are going to be bigger than females on average. Of course, it's going to vary from person to person, but 9 times out of 10, guys have more muscle mass. That's due to a lot of things. They have more testosterone, which allows them to grow more muscle. We don't need to get into the specifics of it for this class, but males have more muscle, which means they have more muscle fibers or muscle cells, which means they have more actin and myosin, and they have more motor units. So they just have the ability to have greater strength, essentially. Okay, and number 4, this is the last one I'll go through right now. The muscle fibers in a motor unit are dispersed throughout a muscle and intermingle with muscle fibers of other motor units. What is the functional significance of such an arrangement? If you haven't done any of the worksheet yet, go ahead and go back up to number 3 and write down the definition, find the definition in your textbook of what a motor unit is, and then come back to this question and see if you can think about what the answer might be for number 4. And I will tell you that we have usually for each muscle, like if I'm talking about my bicep muscle, you probably have a couple nerves that are serving that muscle coming from your spinal cord, but one nerve, if it's kind of coming in on the side of the muscle, another nerve kind of enters the muscle over here. The axons that branch off of that nerve are going to intermingle throughout the entire muscle. So pause the video, go back to that definition, see if you can answer number 4 on your own. But if you have no idea, the significance is essentially like if you damage one of the nerves, your muscle will still work because you have another nerve that is still serving most of the muscle. So we don't want to be one-sided in what part of the muscle we're serving. We want to cross those axons around each other. So if we damage one nerve, we can still use the muscle. So the effects of the damage to a nerve is going to be more widespread, but it also means that we can probably still use the muscle in a greater capacity than we would be able to otherwise. So work on 5 and 6 on your own. If you have issues with those, shoot me an email because I want you to work through this, some of this on your own, but your book has really, really good sections on specifically motor units and all of like the actin and myosin stuff. So I would just start reading through that a little bit and see if you can answer numbers 5 and 6. So I want to go through one final thing. Skeletal muscle anatomy is a little bit complex, not in the sense that it is a complex tissue, because it's really just, the same thing over and over and over again, just this very specific arrangement, but you maybe have never learned about it before. And so the terminology just takes some getting used to. And I can post some good review videos for you if you need some help on that, especially when you get to like the sarcomere stuff and, the sliding of the actin and myosin and how that all works. But let's just talk about it for a quick couple of minutes here. So this entire thing that you're looking at connected to a bone is a whole muscle. Each muscle is made up of these bundles that we call fascicles. And so the fascicles are going to be surrounded by, usually we have some blood vessels in there. There will also be nerves in between here, but you usually don't see those in diagrams. If we pull out one fascicle, we are going to be seeing a bunch of muscle cells. And, I like to always point out, if you see organelles, you're probably looking at the cellular level, because even though muscle cells are weird and they just are filled with a bunch of proteins instead of a lot of cytoplasm, they still have organelles, just like any other cell that you're used to seeing. So we have a muscle, and each muscle is made up of fascicles. Each fascicle contains a bunch of muscle cells or myofibers, and sometimes you'll see it called myocytes. So all of those things are just a muscle cell. The muscle cell is made up of myofibrils, and that's where you have the sarcomeres and all of the actin and myosin. So the covering around the entire muscle is the epimysium. The covering around an individual muscle cell is the endomysium, and the covering around a fascicle, kind of in between the two, is a paramysium. So you'll have to use that terminology for your first question on this lab worksheet. So come back to this picture in your textbook if you need to. It's a really good image, and your book does a really good job of describing these things as well if you need to review that. But let's look at a couple of the microscope slides that we looked at in class for those of you who were not here. And your book has a ton of nice diagrams for this chapter. So if you're ever confused, go through and look at those diagrams, because honestly, they're, one of the most helpful ways for you to kind of review and see if you have a good understanding of how this stuff works and to get a visual of it. So this is showing you that we can have more than one nerve coming into a muscle from the spinal cord. It's possible that you have a few nerves serving each muscle in your body, like the bicep muscle, for instance. But each of those nerves is going to create a motor unit. So each nerve is going to serve a bunch of cells because these axons are going to break off and they're going to become very branched. And you can see that happening right here. But each one is going to terminate at one muscle cell. And so each nerve and all of the muscle cells that it terminates at is going to be called a muscle unit, or a motor unit. So we can have more than one motor unit, and that's why if you have spinal cord damage, maybe your muscle can still work because maybe you just damaged one cell, one nerve and not the other. So the longitudinal section is always when you're looking at like the long tubes, the long cells, and you will see the striations here, and that's because of the arrangement of the sarcomeres with all of the actin and myosin perfectly lined up so it can get ready to contract and shorten the muscle in order to create that tension. In the cells, you will see that there are a lot of nuclei, and you see this mostly in the longitudinal section. If we look at the cross section, we don't see as many because you're just taking like a little slice of it. So like I would take this little teeny tiny section and look down on it. And so of course, you're only going to get maybe just like 2 or 3 nuclei per cell captured in that image. So yes, you should always see nuclei when you're looking at the cellular level. And when you're looking at the longitudinal section, you should always see striations. So that kind of helps orient you to what you're looking at. This is an image that I took in lab of our motor units. So this is a really cool image. You can see the axon coming in from this nerve and it branches a lot. So as it gets kind of more spindly looking, those are just kind of branches off of the initial nerve coming into the cell. Each one of them terminates at a single cell. So each axon ends at one cell, and each cell only has one axon terminating at it because we don't need multiple nervous signals going to the same cell. That's just not necessary. Looking at the cross section of skeletal muscle. So this is a part that can be the most difficult to try to draw. And this is what you're drawing for number 1. So I wanted to orient you to this today. So we have a muscle cell right here, and you can tell it's a muscle cell because you see a nucleus on it. So we have a lot of nuclei, and they're usually kind of on the edge of a cell because they're going to be right under the endomysium, essentially. So they're usually on the outside. And so then the fascicles are going to be kind of just packets of these cells, and it can be more difficult to tell how they're arranged. But like this would be a fascicle. This portion right here would probably be a fascicle. And separation always happens when you're creating a slide. You're taking tissue from a living body and trying to put it on a piece of glass. But this right here would probably be a larger fascicle. And then on the outside, we have the epimysium, which is the outer layer of the muscle. Surrounding each muscle cell would be the endomysium, and surrounding each fascicle would be the paramysium. So you can kind of visualize where those would be. And when you go to actually draw this, you can make your drawing much more simplified if you would like to. You don't need to put this much detail into it. So if you have any other questions on that, let me know. I couldn't get like a really clear picture because our slides are just not the best for this. But if we zoom in even more, you can start to see that each muscle cell is going to be made up of these little kind of darker specks, and they almost look like striations. But you'll be able to even kind of see the pattern of what the myofibrils would look like in all of those cells. So you have the nuclei still. This is just one cell made up of a bunch of myofibrils.